I want to begin by dedicating this talk to my parents. This is my mom, Elsie, and my father, Bob. And this was at the College of Arts and Sciences Distinguished Lecture. And Mom has since passed, but my father's here, and it's his birthday today. So happy birthday, Dad. This is also my lovely wife, Kim, and I'll acknowledge her again later on in the talk. Should I forget to do that, I may need a ride home or a couch to sleep on tonight. Let me also acknowledge my academic parents. This is Professor Nira Ben Jonathan, my PhD advisor, and Nira is here today, so welcome. Thank you very much for coming. And this is my postdoctoral advisor, Professor Mark Whiteman. He does have an excused absence. He's out of the country. Okay, so here's the game plan. I want to give you a little tutorial on dopamine neurobiology so you be prepared for some of the things I'm going to say. I'll go into Parkinson's disease next, drugs of abuse, and then I'll end with monitoring neurochemicals in the human brain. So dopamine neurobiology 101. This is the human brain, and I'm interested in the dopamine neurons that originate here in the midbrain, in the substantia nigra and ventral tegmental area, that ascend and innervate the forebrain in regions called the cortex and the striatum. These dopamine neurons perform important functions such as motor control, cognition, and motivation. And when these dopamine neurons dysfunction, debilitating pathologies occur, such as Parkinson's disease, addiction, and schizophrenia. I'm going to blow up this area of the brain called the striatum, and this is where dopamine synapses are, and these are the synapses that I study. Okay, so this is a synapse between the end of a dopamine neuron and a target cell. There's a little gap in the middle between, this is called the synaptic cleft. At any given time, most of the dopamine is actually inside a dopamine neuron, inside a synaptic vesicle. It's inactive. In order for dopamine to be active, it has to get outside of this terminal. So there's an electrical signal called an action potential that comes down and it releases dopamine into the synaptic cleft. This dopamine can diffuse and act on the target neuron via dopamine receptor, and this is how dopamine alters the electrical activity of a target neuron. And then to shut down the whole process, there are transporters that will take dopamine back up. This is a general mechanism by which neurons communicate with each other. They just use different neurotransmitters. The only little change here for dopamine neurons is that it's an open synapse. So dopamine can actually diffuse far around in the brain and act on neurons whichever have a target neuron. So that's dopamine signaling. The dopamine synapse is an important target of drug action, and I'll be talking a lot about these drugs that act on the dopamine transporter and block uptake. Amphetamine, methamphetamine, cocaine, Wellbutrin is an antidepressant. Ritalin and Adderall are drugs used to treat ADHD. There's a whole class of drugs that act on the dopamine transporter, Haldol or Spiridone, which are antipsychotics used in the treatment of schizophrenia. There's Requip and Mirapex, which are drugs used to treat Parkinson's disease. And then another drug used to treat Parkinson's disease is levodopa or cinnamon, and this activates dopamine synthesis. And I don't show it, but dopamine synthesis also occurs in the dopamine neuron. Okay, so I've been talking about this relationship between action potentials coming in and dopamine coming out, and there's two ways by which dopamine signals other neurons. There's something called tonic signaling, where there's sort of a regular slow volley of action potentials coming in. And this produces something called dopamine tone, sort of a constant level of dopamine in the brain. And the function of this dopamine tone is a gate. When dopamine tone is high, the gate is open and behavior can occur. The other form of signaling is called phasic firing, and you can see these are very rapid bursts of action potentials, and these cause these concentration spikes or dopamine transients. And the function of these signals is learning. And the gate is easy to understand, but learning is a little more difficult. So let me just add one more slide here to my introduction, talk about the role of dopamine in learning. So phasic dopamine signaling is involved in what's called motivated behavior or goal-directed behavior. And I'm going to try to describe this by going back to this old TV show. You remember Let's Make a Deal with Monty Hall? Well, if you recall, there's a segment there where contestants had to choose the right door with the prize behind door number 1, door number 2, and door number 3. And this is actually a good device to understand goal-directed behavior because to meet needs for their welfare and survival, animals must adjust their actions for achieving goals in a changing environment. The prize isn't always behind door number 1. Sometimes it's behind door number 2 or door number 3. Also, animals must predict the outcome of their actions for achieving goals. On the show, the contestants were guessing. Imagine how successful they could be if they had cues that would help them predict the right outcome. So in this context, phasic dopamine signaling assesses reward prediction. Okay, and I'm going to show one more thing on the slide here, and don't get nervous because it's actually really, really simple. Okay, so I'm going to describe the relationship between reward, dopamine, and behavior. If the reward is better than expected, there's an increase in dopamine, and this reinforces behavior. This is positive reinforcement. We as teachers know that. If the reward is as expected or as predicted, there's no change in dopamine, no change in behavior. If the reward is worse than predicted, there's a decrease in dopamine and avoidance of behavior. This is negative reinforcement. We teachers know that too. So the role of this type of dopamine signaling is in reinforcement behavior. Okay, so you can all breathe a collective sigh of relief. This is all the dopamine neurobiology I'm going to show you for this part of the talk. But here's the take-home messages that we'll bring forward. Extracellular dopamine is functional dopamine, and it's controlled by release, uptake, and diffusion. Tonic dopamine functions as a gate to enable behavior, and phasic dopamine functions in reinforcement learning. Let's move on to Parkinson's disease. So James Parkinson gets credit for Parkinson's disease because of a monograph he published in 1817 called An Essay on Shaking Palsy. And he characterized this shaking palsy as involuntary tremulous motion with lessened muscular power, in parts not in action and even when supported, with the propensity to bend the trunk forward and to pass from a walking to a running pace, the senses and the intellects being uninjured. And today this is a largely very accurate view of Parkinson's disease, and we speak of these cardinal symptoms: a resting tremor, a rigidity, akinesia, slow movement or no movement, and postural instability. And here's a picture of what Parkinson calls a propensity to bend the trunk forward in Parkinson's disease. It's very clear that Parkinson's disease is associated with the loss of nigrostriatal dopamine neurons. These were those neurons I was talking about beginning in the substantia nigra of the midbrain and ascending to the striatum of the forebrain. You can see this post-mortem. This is a normal brain, and you see these pigmented cells here. Well, these are the dopamine neurons in the substantia nigra, clearly seen post-mortem. And here's a Parkinson's brain, and you can see a loss of these pigmented cells. You can also see Parkinson's disease in the living brain with imaging. Here's a PET scan, which is imaging dopamine neurons in the striatum. Here's the healthy individual, and here's the Parkinson's sufferer. Here's some other facts about Parkinson's disease. The average age is about 62 years old. However, there's a young onset Parkinson's disease, and this is described as occurring younger than 50 years old, and this occurs in about 5 to 10% of the cases. In terms of the cause or etiology, typical Parkinson's disease is idiopathic. It's of unknown origin. However, there's a strong familiar inheritance for young onset Parkinson's disease, a genetic basis, and several genes have been identified. There's about 500,000 sufferers in the United States alone, which means there are many millions worldwide. There is no cure, but there are treatments in the forms of drugs, as I've mentioned, and later on the talk, I'll talk about a neurosurgical approach for treating Parkinson's disease. Okay, it's very clear that symptoms of Parkinson's disease are related to the loss of dopamine neurons, but it's not a straightforward relationship. And in fact, there is a very long pre-symptomatic stage whereby dopamine neurons are lost, but before the symptoms of Parkinson's disease occur. And this is a remarkable ability of the brain to compensate. And think about this. The brain has lost 4 out of 5 dopamine neurons and still doesn't present with symptoms. And even early on, these symptoms are very, very mild. Experimental evidence suggests that the primary compensatory mechanism is the maintenance of dopamine tone. We know what dopamine tone is. When dopamine tone is high, the gate is open, we get normal behavior. When dopamine tone starts to drop, the gate closes and we get symptoms. So the question is, how is this dopamine maintained, especially when we're losing up to 4 out of 5 dopamine neurons? And the prevailing view is proposed by Michael Zigmond at the University of Pittsburgh. And he too, like me, enjoys these stick figures. It's a problem in the field. But you know this already, okay? I've given you some help. These are 3 dopamine synapses. Here's dopamine packaged. It's released, diffusion, and uptake. And all Michael Zigmond has done is he's added dopamine synthesis and packaging. So you know all about that right now. And here's dopamine tone, which controls target neurons. So this is what Zigmond proposes when 2 out of 3 dopamine neurons are lost. This is the pre-symptomatic phase of Parkinson's disease. There's no presentation of symptoms yet. Also notice that only the dopamine neurons are lost, not the target neurons. So Michael Zigmond proposes that the remaining neurons become hyperactive and release more dopamine, and then this drives dopamine from the innervated regions to the denervated regions, and tone is maintained. And there's also a compensatory increase in synthesis to maintain this compensatory increase in release. And I'm not going to say much about this increase in synthesis. Well, I'm a measurement guy, and when I first read about this model, I thought it's very elegant, very nice, but I was concerned that the measurements of dopamine release maybe were a little tenuous. So I was interested in trying to take better measurements of dopamine release. And there's also an alternative model, right? You could have this normal release and less uptake. So they're equivalent. Okay, so let me describe some experiments we did early on here at ISU. And I won't say much about my approach. It's a microsensor approach. We can implant a small chemical microsensor called a carbon fiber microelectrode that John talked about. At its tip, it's smaller than a human hair. It's very sensitive to measuring dopamine. And we can implant this in a laboratory rat and take measurements of dopamine. Okay, so I'll show you some data and I'll tell you how we're interpreting this. So this is a graph showing our measurement of tone versus innervation level. Red is control, so this is what you'd expect in a normal animal. And then each of these dots are measurements of dopamine in an animal that's had a portion of the dopamine neurons lesioned. And what you can see by this line that describes the data, it's a flat relationship, all right? You can take out dopamine neurons, but tone is maintained. And I'd say this is an example of adaptation, right? Tone is maintained. And we already know this, you know, we're not the first to show this, but this is an example of the maintenance of tone during the preclinical phase of Parkinson's disease. What we can offer to these measurements is going in at each of these points and determining its underlying control by release and uptake. And you get a different relationship when you look at dopamine release. In fact, dopamine release tends to fall off as dopamine neurons are lost. And we would say, this is not evidence that there's a compensation in release, which is against the Zigmond theory. And also release tends to fall off, which is also evidence that there's no compensation. So if you're keeping track here, we have compensation in tone, but without compensation of the mechanisms that determine tone. So as the Apollo astronauts say, Houston, we have a problem. How can this be? And we've proposed a solution. Okay, so picture a bucket, and you got water coming in and water draining out. And as long as the drain and the water flow is the same, the level of water in the bucket doesn't change. I mean, that's steady state, right? That's common sense. And you can really wind down the faucet, just have a little bit of water coming in and close that drain accordingly, and still that water level doesn't change. That's a consequence of steady state. So we think that happens in the brain. Okay, so the brain is a bucket of dopamine, the faucet is release, the drain is uptake. As long as these two rates are matched, dopamine doesn't change. And we predict that release and uptake always change together because they're on the same neuron and they die off at the same rates. And in fact, in our data, they do change the same. So now we realize it's a bit of an odd theory, it may be too simple, and I just want to show you one more piece of data. And here's what you'd expect for dopamine tone across the dopamine denervation range. Maintenance during the preclinical phase and a drop-off during the symptomatic phase. Well, this is a mathematical model that we've developed of dopamine tone, and guess what? We didn't have to cause a compensated increase in release or decrease in uptake. So the experimental and the theoretical evidence suggests that our bucket theory really works. And as scientists and as serious people, we can't use the word bucket theory, so we've come up with this sophisticated name, passive stabilization. But in my heart, it's the bucket theory. Okay, so the key points here is Parkinson's disease is associated with the loss of nigrostriatal dopamine neurons. Reductions in dopamine tone lead to the presentation of cardinal symptoms, and passive mechanisms may normalize dopamine tone during the pre-symptomatic phase. Let's move on to drugs of abuse. Okay, I'm interested in a class of drugs called the amphetamines, and these are drugs that have a structure similar to amphetamine. And I had to do this for John Bauer, he's a chemist, but here's a bunch of chemical structures of the amphetamines. And you'll recognize many of them. Here's amphetamine, and here's methamphetamine, and the chemist will tell you, well, the difference is a methyl group right here. Everything else is identical. That's why this is called methamphetamine. You'll also recognize methylphenidate, which is Ritalin. You'll recognize NDMA, which is ecstasy. There are others. Here's mescaline, which is in peyote. Here's ephedrine and pseudoephedrine. These are used as decongestants. They're also used to make methamphetamine, which is why you can't get them in Tylenol. in NyQuil anymore unless you go up to the counter. And then a couple more here. Here's fenfluramine. This is the fen and fenfan, which is now a discontinued diet suppressant. And here's deprenyl, which is an inhibitor of dopamine breakdown, and it's used as an antidepressant. So this is a large class of diverse drugs. There's also other unique properties about this class called the amphetamines. Some are highly addictive. So for example, amphetamine and related stimulants, such as methamphetamine, are second only to cannabis, marijuana, as the most widely used illicit drugs worldwide, nearly outnumbering cocaine and heroin use combined. Some of these amphetamines are neurotoxic, like methamphetamine. Methamphetamine will kill dopamine neurons. And then some of these amphetamines are clinically useful as cognitive enhancers. For example, Adderall and Ritalin are used in the treatment of ADHD. So it's an interesting class of drugs, and I'm just going to hit on these aspects in the next few slides. So there's a wide variety of drugs of abuse. They have different mechanisms, they have different effects, but they all have a common mechanism. And that common mechanism is they all activate dopamine neurons to generate a hyperdopamine state. That's what all drugs of abuse do. So here's our dopamine neuron. You've seen this before. I've just added the front end, and this is what causes the electrical signals. We know how several of these drugs already work. Amphetamine, methamphetamine, cocaine act to inhibit uptake. There are other drugs such as alcohol, opiates like morphine and heroin, cannabinoids, marijuana, and nicotine that actually drive the electrical firing of these dopamine neurons. And still some of these will actually increase release itself. So this is a well-established idea in the field of drugs of abuse. There's a couple newer ideas. I'm going to call these emergent hypotheses. And one is that drugs of abuse activate phasic signaling. And this is that learning signal I talked about. And this makes a lot of sense, right? Natural rewards activate phasic signaling. Artificial rewards ought to do that. And here's just an example. So these are some microsensor measurements that similar to what we do, but performed by Danita Robinson. And this is in an awake, freely behaving rat. And this is one of those phasic dopamine transients. And this is a control animal. And all of this shown down here, John Bauer will know what this means, is this tells us it's dopamine, okay? That's what the pretty colors are. This is dopamine, this is the phasic signaling. Well, look what happens after ethanol, right? There's a robust activation of phasic signaling, okay? You can see this for cocaine, you can see this for nicotine, you can see this for the cannabinoids, marijuana. Then the second emergent hypothesis is that drugs of abuse hijack reinforcement learning circuits by activating phasic signaling to a greater extent than natural rewards. These are robust changes. And if these drugs are acting stronger than natural rewards, it's easy to see that why they're such great reinforcers and why they hijack behavior. So these are very compelling hypotheses, but they're not a unifying hypothesis because there's a big exception to this rule. And this exception is our friend amphetamine. It acts on dopamine neurons. It causes this hyperdopamine state, but it's thought to act by a very different mechanism and disrupt this phasic signaling. And to cut to the chase, we've been working on this issue for a few years now. We don't think this is true, and I'll just show you some data that we published earlier this year. This is the effect of amphetamine. So just compare amphetamine to ethanol, and they do the same thing. So amphetamine really does act like other drugs of abuse. And so our contribution to this field is that we're bringing amphetamine back into the fold, and therefore we can now propose these as unifying mechanisms for how drugs of abuse act. We're also interested in methamphetamine because it's neurotoxic. For example, human methamphetamine abuse is associated with the loss of nigrostriatal dopamine neurons and behavioral impairment. And here's a PET image comparing the striatum in a healthy individual and the striatum in a methamphetamine abuser. Kind of looks like Parkinson's disease. And that's true. There's a loss of dopamine neurons, not quite to the extent of full-blown Parkinson's disease, but similar to the pre-symptomatic phase. There's also behavioral impairment in the form of timed motor tasks and cognitive tasks, verbal memory tasks. And so we've become interested in this relationship between these cognitive deficits and meth abusers and the loss of dopamine neurons and what the mechanism is. And we know it's not tone, because tone stays high, but it might be a deficit in phasic dopamine signaling. And we've been working on this for several years now, and I'm just going to cut to the chase. And this is the paper that'll be coming out this year. And we can now show that methamphetamine-induced neurotoxicity impairs phasic signaling. And here are some phasic transients in a control animal. Here's a phasic transient in an animal that's received a neurotoxic dose of methamphetamine. You can see it's a little smaller. On average, there's a significant drop in the amplitude of this phasic dopamine signal. Now we're interested in what this means for the target neuron, what this means for behavior, but we're also interested in trying to restore this difference. So can deficits in phasic dopamine signaling and methamphetamine-induced neurotoxicity be restored? And here's some preliminary data saying yes. So this is the effect of levodopa. It's a Parkinson's disease drug. It increases synthesis, which should increase release, which should increase phasic dopamine signaling. And you can see this in a healthy animal, shown here. Here's the effect of L-DOPA on phasic dopamine levels. These are the animals that have received this neurotoxic dose of methamphetamine. And you can see the deficit right here before L-DOPA. But look what happens after L-DOPA. We can bring that level back up to control, and it's sustained for at least a couple hours. Okay, so in summary for this section, drugs of abuse, including amphetamine, activate phasic dopamine signaling. So we now have a unifying hypothesis for drugs of abuse. And what I'd like to add here is that the same mechanism for amphetamine may apply for how Adderall acts in ADHD. So it has wide-ranging implications. Neurotoxic doses of methamphetamine impair phasic dopamine signaling. And to give you an idea of the implications here, there are several conditions associated with the loss of dopamine neurons. Parkinson's disease, methamphetamine neurotoxicity, traumatic brain injury, stroke. All of these have associated cognitive deficits. And these cognitive deficits may be mediated by this deficit in phasic dopamine signaling. So there's some rather broad implications here. And then finally, there are drugs that may restore impaired phasic dopamine signaling, and some of the candidate drugs we're interested in are levodopa, Adderall, Ritalin, and Wellbutrin. And the beauty of these drugs is that they're already being used in humans for various purposes. Okay, so let me end here with monitoring neurochemicals in the human brain. And when we record dopamine in a laboratory rat, we have a wire that's connected between the sensor in the rat and our recording equipment. And we've been trying to replace this wire with a wireless link. Okay? And this is one of the first instruments we've built. It's called Real-time Animal Telemetry, or RAT. RAT to measure dopamine in the rat, of course. It uses Bluetooth. This instrument can be folded up and used as a backpack, and it does measure dopamine. So it's been useful as a prototype or proof of concept. It's established several firsts for the wireless measurement of dopamine, but it is pretty big. I mean, there's a penny here for scale. So more recently, we've been working with engineers at Case Western Reserve University to make a little microchip to do all of this. And just to give you an indication of scale, this microchip can fit on the head of Lincoln on the penny. So considerable reduction in size and more function, to tell you the truth. Well, rat may not be useful for the rat, but it's a suitable size for an intraoperative neurochemical monitoring device. And what I want to show you next are efforts by Mayo Clinic Rochester to take our rat instrument and turn it into a device that can be used in humans for monitoring chemicals during a neurosurgical procedure called deep brain stimulation. And let me just show you the phenomenon of deep brain stimulation, and then I'll tell you more about it. There's some sound here, so I'm hoping it's working.
Can you twist your hand like that? You're trying to twist it.
I'm trying to.
Okay.
I just have no control.
Right.
46-year-old Gary Meredith has had Parkinson's disease for 12 years. Medications don't help him.
Wake up in the middle of the night, you have to go to the bathroom. I'd have to crawl.
As you can see, he can barely walk. Now, watch this.
It's just like somebody takes a switch and turns it off.
That's exactly what happened.
Okay, so that's a deep brain stimulation. What just happened? Well, this patient has a surgery where an electrode has been implanted in the brain. It's been connected to a stimulator that's been implanted in the chest. And what you saw was the physician turning it on wirelessly. And this is causing a really high-frequency train of electrical pulses to go to the stimulating electrode. No one knows quite how it works, but as you can see, it works phenomenally. It's now being used for a variety of pathologies, motor disorders such as Parkinson's disease, general dystonia, essential tremor, as well as neuropsychiatric and other diseases such as obsessive compulsive disorder, depression, epilepsy, and pain. One of the corporate officials of a company that makes these devices called the last 20 years the era of neuropharmacology, and he's calling the next 20 years the era of neuromodulation, right? You're achieving the same thing, control of brain, but in replace of drugs using electrical stimulation. And this is an old number. There's more than 40,000 implants now. Well, here's the concept of a neurosurgeon at Mayo Clinic, Rochester. And what he would like to do is, during the surgery to implant the stimulating electrode, and this is the stereotaxic frame that's used to implant the stimulating electrode, is also to implant a microsensor and then use this wireless device to send information back to a computer and then to monitor neurochemicals during the surgery. These neurochemicals can help in the placement of the stimulating electrode, tell us how DBS is working, and even used to study basic brain biology in a human. And so I'm just going to describe some recent measurements that we were able to publish last year. So this is what's called Thalamic DBS for essential tremor. So this is deep brain stimulation for the motor disorder of essential tremor. It's a little bit different tremor than that in Parkinson's disease. It has a different neurochemical underpinning and a different brain region. And the target of deep brain stimulation for treating essential tremor is the thalamus. So what's shown here, this is the neurosurgeon, Dr. Kendall Lee. This is the patient. The patient is awake. This is that stereotaxic frame. And then here's the wireless device. So the wireless device is connected to a microdrive that can lower a microsensor. It also is the same microdrive that lowers the stimulating electrode. And what you have right here is an X-ray of the stimulating electrode alongside the neurochemical recording electrode in this patient. So that's pretty phenomenal. Okay, so what are we trying to do in this set of patients? Well, we're investigating what's called the honeymoon effect. And this is an incredible phenomenon that there is symptom relief just by physically implanting the stimulating electrode. No stimulation, just physically implanting the stimulating electrode. Happens in about 50% of the cases. And when it happens, it can last for days to weeks. It's a phenomenal mechanism. Okay, so here's an example of this honeymoon effect during DBS of the thalamus for essential tremor. This is a measurement of tremor in that patient. And this is just implanting the stimulating electrode. No electricity. And look at that tremor going away. Okay, the patient is awake during all of this, and what the neurologist does, it has the patient draw before the surgery, and you can see kind of a wobbliness here. And this is the patient right after implanting the stimulating electrode. Look at how nice and tight those lines are. Now, interestingly, this has also been called the microthalamotomy effect, and people used to think you're making a little lesion here. And in fact, lesion surgery used to predate DBS surgery. But we don't think it's a lesion that's causing this. We think neurochemicals are released. And in fact, here's a recording from a microsensor that's been implanted in the thalamus as the stimulating electrode comes on down. And you can see there's a robust release and some sort of chemical here. We think, based upon this evidence, that it's adenosine. And adenosine is another neurotransmitter. And in experimental animals, adenosine can relieve tremor. Okay, so I'm going to try and show you surgery here. And I'll show you what to watch for.
Raise your arms up, sir.
Okay, so there's the surgeon here. This is the patient. The patient has a Wii controller that's measuring the tremor, and the tremors are being reported here. This is the microsensor recording, and at some point, the microsensor will be implanted. Okay, the deep range electrode is implanted here.
Yeah, that's it.
It's now getting smaller.
Oh, nice. Let's get an X-ray, please. Okay, hold your hand down. Hold your hand out, sir. Closer to your nose. Now hold it right there. Look at that. Look. Are you measuring this? Are you getting this? Look at this. Look at his hand.
Okay, so this is tremor relief from just implanting the stimulating electrode and monitoring what we think is adenosine release at the same time. Okay, so the key points here, I do think ISU rules for a lot of reasons, but it's given me a great deal of satisfaction and pride to think that an instrument that developed here at ISU is being used by Mayo and developed into a human monitoring device. And then also human intraoperative neurochemical monitoring is now a reality. So let me thank some people. I feel blessed in many regards. I've had great parents, great mentors. I've had great students at the undergraduate level, graduate level, postdoctoral level, and great colleagues. I would like to thank all of them. The people I'm acknowledging here are the ones who have been most directly related to the research that I've described. And I just want to identify two of the graduate students here, Chris Howard and Dan Covey. They're in the audience. Chris was the student who took those heroic measurements of dopamine in a meth neurotoxicity model. Very difficult. I didn't think it was going to be even possible. And then Dan Covey is leading the charge and trying to figure out exactly how amphetamine works. And we've had a lot of nice funding over the years, and I hope it continues. And then finally, so I'll get that ride home to my lovely wife, Kim. We've been married going on 22 years. She's also the mother of our two beautiful children, Abby and Zeke. And she is also the research technician in my lab. So she runs my lab, and you could say she runs every aspect of my life. Anyway, thank you very much.